So first, I'd like to say thank you very much for being invited to contribute to this workshop today. And we've had two absolutely fantastic sessions on the brain. So my disclaimer, if you like, is that I'm going to bring us down a level and look at the peripheral and spinal circuits that are involved in pain symptoms. Okay, so actually, this doesn't work. So because this is an educational workshop, I thought I'd first start off by giving a kind of brief overview of the circuits. So we know that primary sensory neurons reside in dorsal root ganglia and that their peripheral processes innervate somatic and visceral tissue. And those sensory nerve endings contain an array of transducer receptors and voltage-gated ion channels that detect a broad range of sensory stimuli and trigger the generation of action potentials. And the primary sensory neurons then relay those action potentials to the spinal cord. So in the spinal cord, both pain and tactile information undergoes processing by local excitatory and inhibitory controls, and there's also important descending control from the brain. And following processing, signals are then sent to higher centres that are known to be involved both in the sensory discriminative aspects of pain processing and also the affective emotional aspects, and as we learned last night, are very important for the chronicity and the development of chronic pain. So this somatosensory system that processes pain and also tactile information is essential in order to safely but also richly experience our world. So we know that this somatosensory system is not fixed, we know that it's dynamic, and that following injury, there is plasticity in the system that can drive hypersensitivity. And that hypersensitivity is characterized by the symptoms of hyperalgesia, allodynia, and spontaneous pain. So we know that hyperalgesia is when a painful stimulus is perceived as being more painful, exaggerated pain, whereas allodynia is when a non-painful stimulus is perceived as being painful, if you like, tactile evoked pain. And these symptoms occur following tissue injury, inflammatory pain, and also following nerve injury, neuropathic pain. So in these conditions, there is plasticity within peripheral nerves, termed peripheral sensitization, and there's also plasticity at the level of the spinal cord and brain, termed central sensitization. So we'll look at these in a bit more detail. So we know that following injury, there are interactions between these nerve endings and immune cells. Basically, there are bidirectional neuroimmune interactions, and these very rapidly sensitize these nerve endings. There's nociceptor sensitization, and there's also altered gene expression. And collectively, this is what we term peripheral sensitization. And that drives an ongoing pain input to the spinal cord that induces plasticity within the spinal cord. There's evidence for increased excitatory control, decreased inhibition, and we also know there's altered control from the brain. And bidirectional neuroimmune interactions are also involved in some of these changes. So as a result of this plasticity, there's increased activity in those ascending pathways to higher centres that also induces plasticity in these regions. So that plasticity at spinal and higher centres is termed central sensitization. So first, we'll take a closer look at peripheral plasticity. So this term, nociceptor sensitization, really describes the mechanism responsible for reducing the threshold for activation of a nociceptor and driving an increased response in a nociceptor. So how does this occur? So here we have a nociceptor nerve ending with its transducer receptors and ion channels that are required to transduce sensory stimuli into electrical signals. So following injury, we know that inflammatory mediators are released from damaged tissue cells and also immune cells. And these can both activate the nociceptor ending, but they can also modulate the sensitivity of the ending. And they do this by acting on the receptors that stimulate signaling pathways that drive the phosphorylation and modulation of these ion channels and transducer receptors, and basically make it easier to generate action potentials in the terminals. They lower the threshold for activation. And by modulating ion channels such as HCN2, they can boost the firing rate of the nociceptors and enhance responses. Importantly, these changes take place very rapidly, allowing the somatosensory system to dynamically respond to injury. But there are also longer-term translational and transcriptional regulation of gene expression that can modify nociceptor phenotype and be important for pain persistence. So more recently, there is growing evidence that signals are not just passively transmitted from the periphery to the CNS. There's evidence for regulation, both at the level of the dorsal root ganglion and also at the level of the axon. So using in vivo confocal imaging in the DRG, the Dong group has demonstrated that, or discovered, that there is electrical communication in the dorsal root ganglia. So under normal conditions, a pain stimulus will activate given DRG neurons that then transmit that pain signal to the CNS. But really interestingly, following injury, that pain stimulus will activate that neuron or those DRG neurons that then rapidly activate its neighboring neurons via electrical gap junctions. And you can see this is a really neat way to amplify the pain input to the central nervous system, and this is thought to contribute to hyperalgesia. So as well as electrical communication in the DRG, there is also evidence for GABAergic communication within the DRG. So the machinery required for GABAergic neurotransmission is present in the DRG and is functional. So you may remember that primary sensory neurons are pseudo-unipolar and they form a T-junction. And pain signals must cross this T-junction on their way to the CNS. And interestingly, Nikita Gamper and colleagues have shown that in the presence of GABA, these pain inputs can actually fail to cross that T-junction. And so if you like, GABA in the DRG can inhibit or gate pain input to the CNS. And there's some evidence for a deficit in this gating mechanism in the scenario of injury. So again, this could contribute to hyperalgesia. So now I'd like to focus on axonal regulation and show you some work from my lab. So we're interested in the peripheral nerve relay of pain. And just to remind you that in the nervous system, information is coded both by the number of action potentials and the time intervals between them. So if these action potentials encode this level of pain, if those action potentials are more spread out, that would indicate a smaller pain stimulus, whereas if you have more action potentials, that would signify more pain. Now, interestingly, the pain-sensing C fibres that relay pain signals to the CNS have an interesting property. So when action potentials are triggered in the nerve terminals, they're not just passively transmitted along. Due to a process called activity-dependent slowing, these action potentials actually become progressively more spread out as they are relayed along the nerve. And this is thought to act a bit like a brake on pain input to the CNS. So this activity-dependent slowing is thought of as being a brake on pain input to the CNS. So we can study this very simply in isolated peripheral nerves from rodents. And with a stimulating electrode, we can drive electrical signals. And essentially, we can monitor how long it takes the signal to reach the recording electrode. So here we have one of these C fiber responses. But if we stimulate repetitively, you can see that the response takes progressively longer to reach the recording electrode, and that's illustrated by this shift in the C fiber response. And this example here actually is an example from a male subject, and here is an example from a female subject. And you can see that that shift, that break is less, and that is quantified here. There's less of a shift in the female subjects. So the females, if you like, they have less of a pain break, and that fits with the observation that these female subjects have higher thermal pain sensitivity. So this work is from mice, but we have also observed sex differences in this break and thermal pain sensitivity in the rat also. So what about this pain break in the context of pain conditions? So we have looked at this in relation to diabetes-related nerve pain, and it has been shown that there are raised concentrations of the glycolytic metabolite methylglyoxal in the plasma of diabetic patients who go on to develop neuropathic pain. And interestingly, methylglyoxal post-translationally modifies the voltage-gated sodium channels 1.7 and 1.8 that are involved in this pain signal relay. So we asked, does methylglyoxal alter this break? So in this male example here, methylglyoxal appears to reduce the break, whereas in this female example, we see actually the complete opposite. We see an increase in this break, and that is quantified here. So in the presence of methylglyoxal, there's a reduction in the pain break in males, but an increase in females. And if we systemically administer methylglyoxal to subjects, in males, this drives a thermal hyperalgesia, but in actual fact, in females, it does not. So we see a sex-dependent effect of methylglyoxal on this pain break and behaviours. And we've also observed sex-dependent changes in this break and thermal pain in other preclinical pain models. Okay. So just to summarise this peripheral plasticity section, we've just reminded ourselves that there are bidirectional neuroimmune interactions that sensitise these nerve endings. And as part of the more recent discoveries following injury, there is evidence for novel electrical communication in the DRG and possibly also altered GABAergic communication. And work from my lab has shown that there is an alteration in this pain signal relay in axons. So collectively, this will amplify pain input to the CNS. So now we'll look at central sensitization. So that ongoing pain input will amplify neurotransmission with spinal neurons, and that elevates intracellular calcium levels that will recruit kinases, that will drive rapid post-translational changes as well as long-term translational and transcriptional changes. So if we look at the spinal cord, as I mentioned earlier, these changes manifest as an increase in local excitability, a decrease in local inhibition, and there's also altered descending control. So overall, there is an increase in excitability in the spinal cord. So what I'd like to do now is look at how these changes and that altered sensory processing actually relates to those pain hypersensitivity symptoms. So following a peripheral injury, we know that there are two main zones of hypersensitivity, the injured zone in red, but there's also this surrounding area in blue that is not injured, but also becomes hypersensitive. So hyperalgesia in the injured zone is thought to be due to the peripheral plasticity mechanisms that we've just looked at, whereas hyperalgesia in the surrounding zone is thought to be due to central mechanisms, and allodynia is observed in both regions, and this also is thought to be due to central mechanisms. So following injury or inflammation, we know there's this pain drive to the spinal cord that increases excitability. And it's thought that this enhanced excitability then enables A delta mechanical pain fibers to access circuits and drive mechanical hyperalgesia. And it enables A beta fibers, touch fibers, to access circuits to produce the symptom of mechanical allodynia. So now I'm going to show you some electrophysiology studies where I've explored the circuits involved. So using patch clamp electrophysiology in spinal cord slices, I've recorded from a group of output neurons. And in this preparation, the sensory nerve is left attached so that you can electrically stimulate the incoming sensory input. And using different electrical stimulation intensities and parameters, we can characterize the sensory input to the neurons, whether they're A beta or C, and also determine whether inputs are direct, monosynaptic, or indirect, polysynaptic. So under normal conditions, these output neurons are dominated by direct C fiber input, but they also have some A delta input. But in an inflammatory pain model, they actually have double the amount of A delta input. But interestingly, if you look more closely at the control condition, it turns out that those A delta inputs are actually physically there, they're anatomically present. It's just that they are functionally silent. And they're functionally silent because they only have postsynaptic NMDA receptors, and you need postsynaptic AMPA receptors for a synapse to be functional. And it's likely that in inflammatory pain, these are activated by the delivery of AMPA receptors to the synapse. So using similar approaches, I've also looked at allodynia circuitry. And one of the key features of central sensitization is a disruption of spinal inhibition. In neuropathic pain, this can involve microglia and alterations to anion gradients, and in inflammatory pain, prostaglandin-mediated disrupted inhibition. So if we pharmacologically mimic this disrupted inhibition, this actually unmasks a novel input from A beta fibers to these projection neurons, and that is indirect via spinal neurons. So this means that touch information could now be sent in ascending pathways to the brain that would normally exclusively carry pain signals. So this may be relevant for allodynia. So interestingly, over the last decade, a number of different research groups have begun to identify the neuronal subtypes that are involved in this allodynia circuitry using optogenetic, chemogenetic, and genetic neuronal ablation approaches. And interestingly, overlapping yet distinct allodynia circuits are recruited in inflammatory versus neuropathic pain. For example, somatostatin and V-GLUT3 subtypes are involved in both pain conditions, whereas calretinin and dynorphin are key for inflammatory pain, and these ones are important for neuropathic. And there's also been investigation of the inhibitory neurons that normally would prevent this circuit from activating the output neurons, whose function is compromised in these conditions. And RET and dynorphin subtypes have been implicated in both pain conditions, and pervalbumin for neuropathic pain. So this huge body of work from multiple groups is really nicely summarized in this review. So lastly, I'd like to tell you about some of our more recent work, and if you could read the title, it says post-surgical tactile pain. So in this study, we focused on the A beta touch fibers that provide the tactile drive to these spinal allodynia circuits. So we've used the Brennan rat hindpaw surgical incision model, where under anesthesia, a skin muscle incision is made in the heel region of the glabrous skin of the hindpaw. And in behavioral studies, if you take a paintbrush and just lightly stroke the undersurface of the paw, in the uninjured paw, they really don't respond much at all. But in the days following this surgery, we see a robust pain response. And this peaks kind of post-surgical day 2 or 3 and is resolved by post-surgical day 7. So this kind of nicely models post-surgical tactile pain. So what we've discovered is that there is an altered structure and innervation of the skin in this model. So to explain, if we first look at the kind of pad region of the hindpaw, this has a specialized structure that you can see in tissue sections. So in blue, we have the dermis, and you can see there are protrusions of the dermis into the epidermis, and these protrusions are called dermal papillae. And what they contain, or they house, these specialized tactile nerve endings called Meissner's or tactile corpuscles. And these beautiful structures contain an S100 bulb of lamellar Schwann cells, and it's innervating A beta fiber. And the bigger the bulb, the more the Schwann cells, the greater the tactile sensitivity. But in the control heel region, this structure is not present, and neither is this innervation. But really interestingly, following incision, and that is the incision site here, we have observed the development of dermal protrusions that appear to be oriented towards the incision site. And if we actually look at the density of these protrusions, they're pretty comparable actually to the density of these structures in the control pad, which is specialized to detect fine touch. So not surprisingly, we asked, do these putative dermal papillae contain tactile corpuscles? And so we have immunostained for these structures in both the control pad and incision tissue. And if you look at the tops of these dermal papillae, zoomed in here, you can see the S100 bulb and the innervating nerve fiber. And really interestingly, in the incision tissue, in these putative papillae, we see the same structure. So, and if we look at the density of these tactile corpuscles, by looking at these, quantifying these S100 bulbs, the density of these peaks at the peak of allodynia and is reduced at allodynia resolution. So, following incision, we appear to have the development of these novel tactile corpuscles around the incision site. And during development, these corpuscles require BDNF tract B signaling in order to form, with the skin epidermal cells containing BDNF and the tactile nerve ending, the tract B receptor. So we asked, is this signaling required for these novel tactile corpuscles and this tactile allodynia? So first of all, given we have these novel structures, we looked to see if there was any change in BDNF expression. And with BDNF immunostaining, here's the incision. We can see here in the epidermis, there is an increased immunostaining intensity compared with distal to the incision site, and that is quantified here. So this is raised at post-surgical day 2, but has resolved by post-surgical day 7. And to test the requirement of this signaling, we used a tract B FC chimera to sequester BDNF around the incision site, both on the day of surgery and again on post-surgical day 2. And interestingly, when we look at post-surgical day 3 with behavior, we see a reduction in that tactile evoked pain behaviors. And when we look at the tactile corpuscles, in fact, the density is not altered, but the size of those tactile corpuscles is reduced by about half. And reducing the bulb area, the size of the tactile corpuscles will reduce tactile sensitivity. So what we have shown here is that BDNF tract B signaling is involved in the development of the novel corpuscles and the tactile evoked pain. So just to summarize this section on hyperalgesia and allodynia circuitry, following injury, ongoing pain input increases excitability in the spinal cord. And I've shown you that some of these projection neurons have silent direct A delta inputs that become activated that may be relevant for mechanical hyperalgesia. I've also shown you that there is this A beta drive to the projection neurons that's normally under strong inhibitory control, but that following injury, when that inhibition is compromised and there's a strengthening of excitation, that allodynia circuit is unmasked so that touch information can be carried in these pathways. And that may be relevant for mechanical allodynia. And lastly, I've shown you that in the context of surgery, there appears to be the development of novel tactile corpuscles surrounding the injury site that seems to involve a BDNF tract B mechanism. And we propose that this provides or amplifies the tactile drive to these spinal allodynia circuits. And lastly, I'd just like to thank the lab members, current and past, that have been involved in some of the work that I've presented today. And I'm more than happy to take questions.